Welcome to this explainer. Today, we're diving headfirst into the ICHQ-14 guideline, and our goal is pretty simple. We're going to transform what is honestly a pretty dense regulatory document into a clear, structured roadmap for building truly robust analytical methods. If you work anywhere in pharmaceutical analysis, or really any scientific field that relies on precise measurements, this is absolutely crucial. It's the foundational blueprint you've been looking for.
Okay, let's quickly run through our agenda for today so you know where we're headed. First, the problem with inconsistency. Second, what exactly is ICHQ-14? Third, building a strong foundation. Fourth, minimal versus enhanced approaches. And finally, number 5, managing the method lifecycle. Let's jump right in.
All right, part 1, the problem with inconsistency. To really appreciate why this guideline even exists in the first place, we have to look back at the headaches laboratories have been dealing with for years. Think about it like making your morning tea. If you're constantly changing the amount of tea leaves, tweaking the milk, or messing with the boiling time every single day, well, your tea's going to taste completely different every time, right? That exact same logic applies to pharmaceutical analysis when methods just aren't reliable. If a method running on an HPLC is sensitive to tiny, unmonitored little tweaks, your results are going to bounce all over the place. The ultimate goal is that a method should give you the exact same result no matter who is running it, when they're running it, or what lab they happen to be standing in.
And honestly, this perfectly captures the old way of doing things. It was this tedious trial and error cycle that most analysts are way too familiar with. You prep your mobile phase, you run the HPLC, and the peak looks awful. So what do? You tweak the pH and run it again. Still looks bad, you swap out the column and run it again. You just stay stuck in this blind loop until you finally get a chromatograph you can use. But this approach, it led to frequent method failures, a nightmare when trying to transfer methods between labs, and, worst of all, a total lack of scientific understanding as to why the method actually worked in the end.
Which brings us to part 2, what is ICHQ-14? We desperately needed a better way, and enter our hero, ICHQ-14. This is where the industry standard takes a massive leap forward. Published by the International Council for Harmonization, it completely flips the script. Instead of just doing a validation right at the very end of the process and kind of crossing your fingers that it passes, this guideline forces us to shift our focus. It demands that we build quality into the method from day one, developing the procedure scientifically so it's inherently robust, reliable, and actually suitable for what it's intended to do. Moving right along to part 3, building a strong foundation. So how do we actually build this scientific robustness? Well, it starts right here. The source material explicitly calls this the heart of ICHQ-14, the ATP, or Analytical Target Profile. Before you even touch a beaker or turn on an instrument, you have to ask one foundational question. What does this analytical method actually need to achieve? The ATP is basically a perspective summary. It lays out exactly what you're measuring and the strict criteria it has to meet. And the cool part? Your ATP actually dictates your choice of technology. You don't just grab an instrument because it's sitting there in the lab, you choose it because it specifically fulfills your performance requirements. I like to think of ICHQ-14 like building a really solid house. The ATP, that is your architectural blueprint. From there, knowledge gathering is like surveying the land to really understand the environment you're building on. Then, your risk assessment comes in to identify any structural weak points before you even pour the concrete. Things like analytical quality by design and design of experiments, those are your engineering tools that finally get you away from all that guesswork. And wrapping it all up, your control strategy is your long-term maintenance plan, making sure the house stays standing strong for years. Next up, part 4, minimal versus enhanced approaches. Okay, so the foundation is poured, now you actually have a choice in how you apply these principles, because the guideline offers 2 pathways, a minimal approach and an enhanced approach. Now, the minimal approach gives you fixed analytical procedure parameters and strict set points. And hey, that's totally acceptable, but it can be a little rigid. The enhanced approach, though, that is where the magic happens. It gives you a profound, systematic understanding of exactly how your parameters impact performance. Sure, it takes more upfront work. You're doing uni or multivariate experiments and modeling, but the payoff is you get to define much wider operating ranges, meaning your lab gains incredible flexibility because you actually understand the underlying science of your boundaries. Which leads us perfectly into our final section, part 5, managing the method lifecycle. All that enhanced flexibility we just talked about, it pays off big time over the lifespan of a pharmaceutical product. This isn't just a one-and-done deal.
The analytical procedure lifecycle is deeply integrated with product development. It's a continuous, dynamic loop. Prior knowledge feeds directly into your risk assessment, which helps you define your control strategy, which then moves into validation, and finally into routine use and continuous improvement. It proves that developing a procedure isn't just a straight line. It's this ongoing cycle of gathering knowledge, validating, and monitoring. So when changes inevitably happen, they're handled with science, not panic. And speaking of managing change, the guideline gives us an incredibly logical risk assessment pathway to define what are called established conditions, or ECs. Basically, you evaluate the risk of a prospective change. Is it high, medium, or low risk? And that determines how you report it to regulators. If you've done the upfront work using the enhanced approach, you can actually justify having fewer, less rigid ECs. This is a huge deal. It means you establish very clear regulatory boundaries from the start, allowing you to continually improve the method without triggering a massive, expensive, time-sucking revalidation every time a tiny parameter shifts.
To tie this all together, when you compare ICH-QQ14 with the older ICH-Q2 guideline, you realize they aren't competing. They are perfectly complementary. They are literally two halves of the same whole. Q14 is all about the method development, building the scientific understanding, assessing the risk, and optimizing. Q2 takes care of the method validation, confirming the performance and hitting those strict acceptance criteria. Going back to our house analogy for a second, if Q14 is the process of building the house scientifically so it doesn't collapse, Q2 is the final structural inspection that legally certifies, yes, this is safe and fit to use.
I really hope this explainer has shown you why shifting to this quality by design approach is a total game changer. It saves time, dramatically reduces regulatory friction, and honestly, it just results in much better science. So I'll leave you with this final thought to chew on. Take a look at your own lab. Are your analytical methods built on the shifting sands of guesswork and trial and error, or are they built on the truly robust, scientifically understood bedrock of ICH-Q14? Thanks so much for joining me today, and keep exploring the science behind the methods.